Hello everyone and welcome to the presentation on retroviruses. In the previous video, we had discussed about the lytic and the lysogenic cycles, and we had also learned that there is a special type of viruses known as the retroviruses which do not show any type of these following ways of replication. They neither show the lytic cycle to reproduce themselves nor the lysogenic cycle. And here is a video to explain how they reproduce. So to understand well how they reproduce, we need to understand two things. Here in the screen you can see some the DNA, RNA and protein written, and they are joined by some kind of arrows. So what does this mean? This actually refers to how the DNA can be converted to protein in some cases. Most of the cases how the DNA can be converted to RNA and the RNA can be converted to proteins, and also the DNA can be converted to DNA. So they have the special names. What are the special names? When the DNA is converted to DNA, we call it replication, or more specifically the DNA replication, and it occurs in almost all the cells. Okay, it is almost all the it occurs in almost all the cells, and it may be it is occurring, I can guarantee you that it is occurring in your body just now, because there are millions of cells in our body, and there is a high probability that it may be occurring in one of the cell or more than one of the cell just now. So this process of making or replicating the DNA is called DNA replication. Sounds good, right? DNA replication. Replication. Alright. Then the second thing is that the conversion of DNA to RNA, it is known as transcription. So actually we convert DNA to RNA to convert it and ultimately to proteins. So DNA to RNA conversion is an intermediate step of converting it to proteins. So we need to encode our DNA. Our DNA has all the information of making all types of proteins we have in our body. So to make those proteins, we need to convert it first to RNA in our body, we are speaking, and then we need to convert it to protein. So that process of converting the DNA to RNA is known as the transcription. So let us write down there. This process is called, where's my text box? Transcription. Transcription. Okay. Now the ultimate conversion of RNA to protein is called the translation process, and there are ribosomes. If you know, they are carried out by ribosomes. They put together all the amino acids, but that's a different thing. We will talk about them in a different video. There is a huge lot of process doing this conversion of DNA to RNA, RNA to protein. There are a variety of things involved in there, so it is not so easy, but we will discuss them in some other video. Till now it is sufficient for you to know this information. The conversion of RNA to protein is translation. Okay, so these are the three things that normally happens every time in our cells. Okay, the conversion of DNA to DNA, that is DNA replication, DNA to RNA, transcription, and the RNA to protein, that is translation. But in some special cases, some other things also happen, and these are represented by this dotted arrows. And in the case of retroviruses, the conversion of RNA to DNA occurs, and this is also given a special name which is not so fancy, as you will understand if you will hear it. It is called the reverse transcription. So why it is called reverse transcription, you may have made a guess, as the conversion of DNA to RNA is called transcription. It is the reverse of it, right? So it is called reverse transcription. Reverse transcription. Pretty good. And these type of processes, as I told, do not occur in every type of cell. These occur in some special cells. Alright. Now there are other type of processes also. The conversion of RNA into RNA, that is RNA replication. This also occurs in some special cases, and some special cases DNA can also be converted to the protein. So these are, and this whole thing, this whole diagram, as you can see, is known as the central dogma of molecular biology. We will study about more detail about this in some later video. And you can see another thing in your screen. This one is a diagram, is a picture, you can say, of the HIV virus. And this picture I have sticked over here because I wanted to show you that when the virus enters the cell through the receptor mediated endocytosis, as I told in the previous video, there are some receptors in the cell and also there are some receptors in the virus itself. So these receptors connect and these receptors make some signals which the cell interprets and it takes in the virus. So these receptors are there in the virus also. Okay, till now alright. Now let us jump into the replication of retroviruses. How do retroviruses replicate? Let us see. So I have drawn a few steps over here because these steps are very common. This happens in every time because always there will be a virus outside and there will be a cell. The virus will try to gain entry into the cell and the virus membrane will have to dissolve for the genetic material to do its action. Alright, so you can see these three steps over there. So this is always there, but in the case of retroviruses there are three specific proteins in the viruses. Alright, so let's make the brush. There are three specific proteins in case of the retroviruses and I will tell you the names in a second. First let me make the proteins. Here are three specific proteins. I have made them in different colors and they are known, the three specific proteins that help retroviruses to replicate are first reverse transcriptase, second is the integrase, and the third is the protease. We will look at its functions as we proceed further into the mechanism of the replication. So the three proteins, let me make it every time. I have made the virus picture and let me also take care that I put the correct colors. Okay, so there are the three proteins. Here also the three proteins will be there. They will not get dissolved. That is the thing, because if they get dissolved then they have no use only of being there. So they will do some pretty good action while replication of the virus. Now, as I told in the previous video, if I have paid attention and watched the previous video, you will notice that I have told that the retroviruses replication process is much like the lysogenic cycle at the beginning. So this genetic material of the RNA, this genetic material, that is the single stranded RNA, will have to attach to the genetic material of the cell. I just now remembered that I forgot to tell you one thing. That is a very unique feature of retroviruses, and let me write it down over here. Okay, so what is the unique feature of retroviruses? Retroviruses are, I wanted to tell you that retroviruses is a completely different family of viruses. And what is the unique feature of the retroviruses? Retroviruses, let me choose the different color, black will be do it. Okay, retroviruses or retroviruses, viruses are always single-stranded RNA, single-stranded RNA viruses. Okay, and another feature is that they always, retroviruses always have an envelope outside them, envelope outside them. So these are very peculiar features of the retroviruses. Although you cannot say, this is a common misconception, all RNA viruses are not retroviruses, but all retroviruses must have to be RNA viruses. So please pay attention to this point. Many people just say that if the virus is RNA virus, then it must be retrovirus, but it is not the fact. And the same thing applies here. All enveloped viruses are not always retroviruses, but retroviruses always have an envelope. So as I have made the 3 proteins, and also these 3 proteins that I told, the reverse transcriptase, the integrase and the protease are also present in the retroviruses, but this is not a very common feature, so I am not writing that over there, but that is also a unique feature. Alright, so if they always have an envelope, so let me make a envelope outside them. Alright, pretty good. We have not entered till now in the process how they replicate. It is just the beginning. Okay, I don't want to make the okay, there will be no envelope over there. And retroviruses, in this case I have made retroviruses enter to the receptor mediated endocytosis, but it could have entered through the process of fusion of the cell membrane and the envelope. That could also be possible. Now what is the next step? Now these 3 proteins I have drawn. One of these 3 proteins will be in action, and the protein that will do the action now is the reverse transcriptase. Alright, so here's our arrow, and now we will start to dive into the process of the replication of retroviruses. So pay attention. Okay, so here's our cell, and now we have the our single stranded RNA of the retrovirus. What will this reverse transcription transcriptase enzyme do? Transcriptase protein will do is that it will reverse transcript the RNA into DNA. As you can see, I have told this because of that only, that the process in which the RNA is converted to DNA is called the reverse transcription, and the reverse transcriptase will do the same. It will convert the RNA of the retrovirus to DNA. So now from a single stranded RNA, it will now be a double stranded DNA. So let me make this, and then I will explain why it was necessary to convert it to a double stranded DNA. Till that time you can think. Okay, so let me make it as quickly as I can. This is a double stranded DNA, and there will be the cell's nucleus, and there will be the cell's genetic material as always, and this will be DNA. Alright, retroviruses normally attack the cells which have the DNA as their genetic material. This can give you a hint, and this conversion process from the single stranded RNA to the double stranded DNA was done by, as I am always telling, reverse transcriptase. Alright, now time up. I am going to tell you why this is done, why the conversion of single stranded RNA to double stranded DNA is done. As you know, in the if you don't know, but I have given you already a hint that now the lysogenic cycle processes will be done. So the DNA will be ultimately attached to the cell's DNA. So for an RNA to be attached to the cell's DNA, it is very impossible. At least it is very rare that an RNA is attached to the DNA of the cell. The cell will be able to know. The cell will just take the steps to prevent that from happening. So to prevent the cell from knowing that I am here and I am going to attach to the DNA of your genetic material, what they do, what the virus has a plan that it will make its genetic material same as that of the genetic material of the cell, so that it doesn't come to know when it attaches with the genetic material of the cell. Okay, so I hope you got the point. Okay, so that's why the reverse transcriptase has made the single stranded RNA to the double stranded DNA. Now what will happen? Now another enzyme or another protein, enzymes are protein actually, okay, another protein will come into action. This is called the integrase protein, and what will it do? It will actually, the integrase protein will transfer or take the double stranded DNA that has been formed and take it inside the nucleus. Okay, so now let me make the cell again. There's the cell. I will make the nucleus a little larger because it will be difficult to make so many DNAs inside the small nucleus, so I will make it a larger like that. But the nucleus size doesn't expand like that, so don't think like that. Okay, just joking. And then there will be the virus genetic material, the DNA. Let me take care that I make the double strands, and the cell genetic material, the DNA. There's both the genetic materials. Now what they do, the same protein, the integrase protein, will help the virus genetic material get or attach with the cell genetic material. How will they do it? It will just cut off two terminal portions of the virus genetic material. Okay, so they will cut off these two terminal portions, and as there were some base pairs over here and there was some hydrogen bonding between these base pairs, if you don't have this idea, let me tell you that there are some base pairs like A, T, C, G, and there are the hydrogen bonding between the base pairs. So at the terminal region also there were some base pairs and there were the hydrogen bonding, and now the base pairs are not there, so the hydrogen bonding is not done. So there will be this part, due to the hydrogen bonding, there is some potential of having a hydrogen bonding done in these parts. So if the hydrogen bonding is not there, in simple language, these parts will become sticky. Alright, so it has the ability to do the hydrogen bonding, and now when it becomes a sticky, it will stick and do the hydrogen bonding with this DNA of the cell, and like that it will be able to attach itself with the cell's DNA. Alright, so what is the next step? Where is my arrow? Yes, there. Hope you get the point. Now the next step will be that I have chosen white as the color. Alright, now the next step will be, there will be a cell which I am drawing in black, and there will be the nucleus, larger nucleus, which I must have drawn in brown, so let me draw in brown. I am thinking that the video size will be very longer, as some of my previous videos are becoming, but I have to explain the whole process in one video only, because expanding the video making more videos will be useless. Alright, so there will be the genetic material of the virus will stick with the genetic material of the cell. Alright, till now well and good. Okay, now let me complete my diagram. But this time apart, means unlike the lysogenic cycle, when the genetic material of the virus attaches with the genetic material of the cell, it attaches with a repressor gene, right? I had told in the previous video, if you were paying attention, that there was a repressor gene in the genetic material of the virus, and this repressor gene helped the genetic material of the virus not to be understood that it is there by the genetic material of the cell, by the cell. So when it attached in the lysogenic cycle, the virus cell could not know that it has attached. But this time the genetic material of the cell, or the genetic material of the virus, too difficult saying genetic material of the cell, genetic material of the virus, anyways, genetic material of the virus will not have this type of repressor genes. Alright, now therefore this time it will be actively transcripted and translated. Alright, I have used two terms, transcripted and translated. Do you remember these two terms? If you don't know, let us come back to this central dogma of molecular biology. Now, since there is no genetic material of the virus is not repressed, so this will be actively transcripted. So the DNA of the virus will be again converted to RNA of the virus. but this time there will be many RNA of the virus. Every time the cell transcribes its own DNA to make some of its own protein, it will also transcribe the cell's RNA or the cell's or the viral DNA, and then make some to in that process. So let me tell it again. Every time the cell transcribes or transcripts its own DNA to RNA and makes some of its own proteins, it will also in the process transcript the viral DNA which has attached to itself. I hope it is clear now. So in this process, some of the viral DNA will be converted into viral RNA. Alright, so that is the process of transcription, and then it will also be able to translate itself. So the DNA, the viral DNA which has converted into viral RNA, will use the cell's machinery to convert itself into viral proteins, and then again these proteins will assemble and make the more viruses. Okay, but remember, what is the difference here? Difference is that this genetic material does not have to separate with the cells from the cell's genetic material to make more and more viruses. This is an active process. In this process, the virus genetic material is attached to the cell's genetic material, and this process will keep on happening, and the viruses will keep on escaping the cells. So they have the retroviruses have gained permanent control over the cell, we can say like that. Okay, they have gained permanent control. Now the genetic material of the virus will be there forever until the cell is completely cell membrane is completely weared off by the viruses escaping from the cell. It will be there, and it will keep on making the viruses. No such thing of the cell coming to know that the viral genetic material is there, and it's no such thing like the cell trying to repair itself and separating the virus genetic material. No, all the things are not there. It is there, and the cell doesn't know. Cell is thinking that it is its own genetic material. Hope the point is clear. Okay, now what will happen? Now these things will be translated and transcribed. So in the next step, let me make the cell. Okay, there will be the cell. It will become a little small. Okay, because the viruses are escaping and taking a portion of the cytoplasm out, or cytoplasm not, taking a portion of its cell membrane out of it, and viruses are escaping and affecting other cells. So the cell membrane will reduce, and there will be many viruses which have been generated till now by this process of transcription and translation. So many types of viruses, don't care about the shape, but many viruses have been generated. Some of it will be escaping, fusing with the membrane, they have the envelope, and escaping outside with a part of the cytoplasm or with a part of the cell membrane with them. Okay, they will be escaping, and when they mature, or the protein, that is the protease enzyme which they have with them, it will help to remove this part of cytoplasm if they require, and make this part of cytoplasm into an envelope. So the protease will come handy over there. And let me draw the nucleus also in the cell. So there will be the nucleus, and still now in the cell, which I was going to say, the cell will still now have the virus DNA and the cell's DNA. Cell will still now have it, and it will be continuously transcripting into RNA, translating into protein. Continuously this process will continue. Okay, so it will still now have, although the viruses are going on making, the process of making viruses is going on, this genetic material will still now be there. That is the major difference between the lysogenic and the retrovirus replication processes. Now the cell membrane will shrink, and the cell will just become smaller and smaller to be nothing, and then the cell will die off eventually. Okay, so I have just skipped one step over here. The process of transcription and translation will be done, so let me show it over here only. So after it attaches itself, it will make more mRNAs, means sorry, it will make more viral RNAs also, along with making the cells RNA also it will make, and the viral RNA it will also make. Alright, so it will make more viral RNA also, and this viral RNA will use the ribosomes which will be present in the cells, and then make viral proteins out of it. Okay, so it will make long chains. Which color to choose? Okay, this color will be fine. Long chains of viral proteins will be made by it, and these long chains of viral proteins are not very much useful, so the enzyme protease will be used here also to cut off parts of the long chains of viral proteins and make it into small viral proteins which can be assembled. So cut off the useful parts of the viral proteins, like you cut a picture out of a white blank page. So that's how the protease enzyme will cut off the useful parts of the viral proteins and then join those parts to form the capsid layers and other proteins, like the proteins that had been there already. That is, it will form more such reverse transcriptase, it will form more such protease, and it will form more such sorry, it will form more such, what is the other enzyme, forget the names, yes, integrase. For more such integrases, they are all proteins, right? So they will form more such proteins. All the proteins which are there in the virus, they will be synthesized and the viruses will escape. So I have not drawn any proteins in these viruses which are escaping, so they will also have the proteins, all types of proteins, and they will be individual viruses, so they will be similar to the parent virus, right? Sorry, I have to drink water after making this video. Okay, so like that, I hope you get the general idea, and like that the virus will reproduce. So I hope you get what is the difference between the lytic and lysogenic cycle and this type of reproduction in viruses. Alright, so that was it. So, and to connect the thing, after this happens, it will again it will again go on to attack another cell, so it is again a cycle. This thing will again happen. So once it gains entry into the cell, it takes control over the cell permanently and keeps on making its copies. So I hope you found this video helpful. Like it if you found it helpful, and subscribe for more. Thank you.